Hello, everyone, and welcome to today's webinar on energy-filtered 4D STEM and its applications. My name is Anna Paksad. I am the Imaging and Diffraction Product Manager here at Gatan. Our invited speakers today are Dr. Colin Office and Dr. Stephanie Ribbett from NSEM. Colin is a staff scientist at NSEM, part of the Molecular Foundry at Lawrence Berkeley Lab, running both a user program and research group focused on methods, algorithms, and codes for simulation, analysis, and instrument design for TEM. He has received the DOE Early Career Award in 2018 and was awarded the Burton Medal for the Microscopy Society of America in 2022. He is the project leader for the prismatic STEM simulation and Pi4D-STEM open source codes. Stephanie is a postdoctoral researcher, also working at NSEM, which is a part of the Molecular Foundry at Berkeley Labs. She works on 4D STEM development and is focused on phase retrieval techniques. Before we get started, if you have any questions during the presentation, please use the question pane to submit them. Issues regarding connectivity and the webinar will be addressed immediately, and questions for the speakers will be answered at the end of the presentation. Let's get started with an introduction to Gatan's EASY technology. EASY is Gatan's exclusive technology for the most efficient and effective workflows for advanced STEM experiments. EASY combines multiple STEM techniques, it synchronizes STEM detector signals seamlessly, and it links STEM datasets. Let's look at each of these and see how EASY combines, synchronizes, and links STEM experiments. As you know, collecting data only from one type of detector usually does not give sufficient information about the sample you are studying. With EASY, you can simply combine different types of STEM signals in digital micrograph software in order to get a full picture of the sample that is being analyzed in the electron microscope. Here is an example where we have combined EELS and EDS using the EASY technology. As you know, when you're collecting EELS spectrum image data sets, in order to get high resolution energy edges, you might have to select a dispersion which does not allow covering a very large range of energies on the detector that you're using. If you combine now EELS with EDS and collect the data sets simultaneously, you can extend the energy range. As you can see here, we have silicon, nitrogen, titanium, and oxygen mapped using EELS spectrum, and aluminum and tungsten mapped using EDS. And here on the right-hand side, we have the combined EELS and EDS map for a full picture of this 3D NAND device. Combining signals using the EASY technology is as easy as 1, 2, 3. You simply select the types of signals that you want to combine, EELS and EDS in this case. You specify your data acquisition parameters in the SI acquisition pane, and you click capture, and then EASY simply combines the signals for you. Synchronization is very important and required if you want to take advantage of the highest throughput of the STEM detectors that you're using without sacrificing data integrity. With EASY technology, you simply select the signals that you want to synchronize, and EASY does the synchronization for you. You don't have to worry about anything. Here is another applications example. This is a multimodal, multidimensional in situ spectrum image data acquisition, which requires repeated synchronization between different detectors and sequential mode changes on the GIF. Combining EELS, EDS, and 4D-STEM and collecting data as you're stimulating the sample. Easy Python scripting enables and simplifies such a complicated multimodal, multidimensional data acquisition via customization and batch sequencing of the existing workflows in digital micrograph software for STEM spectrum imaging. Easy also allows you to collect temporally and spatially linked 3D, 4D, and 5D, even 5D STEM spectrum image data sets. And you do all of this from within digital micrograph. All the data that are being acquired are linked, and all the metadata are stored with your spectrum image data cubes. This means that you can study chemical, compositional, morphological, and structure function properties of your material with the greatest degree of confidence. Easy not only allows you to collect data sets that are linked, it provides tools within your easy reach in digital micrograph software that allows you to process such linked data sets. This is a really nice example where EELS and 4D STEM data sets were collected using easy technology on this material. Using the 4D STEM data, our application scientists identified particles or crystallites in the sample. And then because these data sets are spatially linked, he was able to go exactly using the picker tool to a specific particle that he was interested, and then look at the EELS spectra from the exact positions in the EELS spectrum image data sets to study the chemical properties of the material at those crystallites. So as I said, easy technology delivers the most effective and efficient workflows for advanced STEM experiments via combining multiple STEM techniques, synchronizing STEM detector signals, and linking STEM data sets so that you get the best, highest quality data sets in the most efficient way. Now I'm gonna give the floor to Colin, and he's going to tell us how easy technology combined with the K3 and the GIF continuum allows energy filtered 4D STEM data acquisitions. Please, Colin.
All right, thank you so much, Anna, for the kind invitation to speak and the introduction. So I'm just going to jump right into it with some application examples here for energy filtered 4D STEM. So right before I get started, though, as Anna mentioned, we are an open access user facility, the Molecular Foundry, of which NCEM, the National Center for Electron Microscopy, is one of the departments. And so if you see anything in either my presentation or Steph's presentation that you're interested in, please feel free to drop us a line either for experiments, analysis, or simulation. So STEM, as Anna mentioned, is an extremely flexible technique. And so you can do all sorts of experiments, including imaging your sample, diffraction space measurements. If you have a spectrometer, you can do either X-ray chemistry measurements or various complicated electron energy loss, EELS measurements. And of course, you can even tilt your sample or have a time-based experiment, as Anna mentioned, and combine it with any of these operating modes. And so the topic of today's presentations here are combining diffraction and spectroscopy. So I'm not going to show any super fancy spectroscopic results. Mostly what I'm going to be focused on is diffraction experiments where we place our camera behind the spectrometer so that we can do energy filtered diffraction experiments. So just a very brief refresher on 4D STEM. In STEM, we typically scan our converged probe across the sample, and a conventional detector like this ADF detector would catch the scattered electrons, a single pixel per probe position. But using our K3 behind the continuum spectrometer, we can record a full image of the diffraction pattern at every probe position. So we take a step, record a pattern, take a step, record a pattern. Typically, this means that we're going to have a two-dimensional grid of probe positions with a two-dimensional image of the diffraction pattern in the back focal plane at every probe position. And that gives this family of techniques their name, 4D-STEM, four-dimensional data sets are what is produced here. So if I just put in some quick toy models here of what nanobeam 4D-STEM looks like, in a polycrystalline sample, you essentially get a two-dimensional Bragg diffraction pattern at every probe position. And I'm going to show examples of the different material science properties we can measure from these diffraction patterns. Similarly, in amorphous materials, if I randomly displace all the atoms on the right-hand side of this toy model and scan through it, you can see our Bragg diffraction pattern is replaced by characteristic amorphous halos. This is because the structure factor of this material has no translational symmetry, and thus you get radially symmetric patterns here. So our 4D-STEM experiments, Anna already sort of demonstrated how we record them using the Gatan STEMX setup here. What I'm going to be mostly showing is examples of different material science properties that we measure, and that's done using our Pi4D-STEM open source Python toolkit for 4D-STEM analysis. So I just want to briefly throw up this website, tinyurl.com/pi4dstem. This has some Google Colab examples. If anybody wants to check out these measurements here, you can run them in your browser without needing to download any code or any data sets. So inelastic scattering, of course, has many, many, many different things you can measure with it. For example, in the Zachman paper, they did chemistry of battery interface materials. In this Yan paper here, excess carbon monoxide was measured at the surface of a catalytic nanoparticle here using eels. If you go down to lower energy regions here and collect a spread of both energy and momentum, this beautiful work by Senga maps out the momentum energy distribution in graphene and compares it directly to DFT. And there's been a lot of good low loss studies. This is another example, the Yan paper around the stacking fault, which was actually mapping phonon modes, so extremely low energy signals, very close to the zero loss beam around a spatially localized defect. And then this is another example from a Brunet paper showing momentum resolved energy loss here, in this case in stacked graphene layers and being able to separate the pi and sigma modes here. So these are all very interesting, but in the context of diffraction though, inelastic scattering typically means an unwanted background signal. So in this Bayer paper here, they recorded diffraction patterns of silicon using the spectrometer to either select the zero loss beam or the first order plasmon loss here at different thicknesses. So I'm just going to show their zoomed in image here on a logarithmic intensity scale. And in particular, I want to call your attention to the right-hand side panel and compare the lower left, the unfiltered image, against the plasmon loss in the upper left and the zero loss image in the upper right. So typically in elastic scattering, what we want is to do detailed measurements of these intensity distributions. And if you compare the zero loss and the plasmon loss intensities, they are very similar, but not identical. And so if you just add them all together, you get unwanted blurring and background signals of your pattern, as you can see in the lower left, the unfiltered panel there. So I think this is a really good example just showing how, especially at low scattering angles, the plasmon losses can produce unwanted background signals. So I'm going to give one quick simulated example before I jump into experiments here. This is an amorphous tantalum sample, and we simulated it using AbTem multi-slice simulations. These coordinates I got from Jun Ding and Mark Asta. And so the way that these amorphous materials are typically characterized, at least on short length scales, is we construct a probability distribution as a function of the bond length between every pair of atoms. So if I start at a single atom, go out to a given radius in real space, what I want to know is the probability distribution inside every spherical shell around each atom and then average it together over the material. So if I just compute the ground truth pair distribution function, or PDF of this material, you can see there's essentially no atoms within the 2.5 angstrom exclusion shell. And then there's a strongly peaked signal that's the first coordination shell at about 3 angstroms. And then you get your sort of oscillating distribution that asymptotically approaches a constant value, because as I mentioned, this material has no long-range translational symmetry. So this is the thing we want to measure with 4D-STEM. So what I've shown here is simulations from Steph. This is the elastic signal only of the material. And then this is the same signal, but I've constructed a synthetic inelastic background and just added it to the pattern here. So I haven't done any plasmon blurring or multiple scattering of the beam. This is quite a thin ideal sample. All we're comparing here is with and without the inelastic background, which, as I mentioned before, for plasmons is peaked around low scattering angles. So if I do a radial integral and plot this on the logarithmic intensity scale here, if you compare these two patterns at high scattering angles, they're almost identical. It's really only at low scattering angles that you're going to get a significant difference. And so when we want to compute the paired distribution function, the first step is to calculate the reduced structure factor. We fit the single atom scattering to this pattern and then subtract it from our diffraction radial average and then normalize by it. So we divide by it as well. And so I've drawn a dashed line here to show the zero value, just to show you that the patterns are nearly identical except at low scattering angles as we approach the origin or the zero loss beam here. So if I use what's called a sign transformation on this structure, we recover the paired distribution function or the reduced paired distribution function. And with a bit of scaling, we can compute the actual paired distribution function. So if I look at the elastic scattering pattern, it looks very good. We have our asymptotic zero distribution as we approach r equals 0. And then we have all of our ringing sort of first coordination shells, second, third, fourth, etc. coordination shells. But if I compare that to the simulation with the synthetic inelastic background, you can see that we've lost about a factor of 2 of our peak to valley signal here, and our oscillations are also not quantitatively correct. So comparing that to the original ground truth distribution, you can see the effect of energy filtering here. It's extremely important if you want to do quantitative studies where you're using your diffraction intensities. Without energy filtering, we just can't make accurate measurements like PDF measurements here. So the other question I should point out here is, when do you need energy filtered diffraction? So here's an experimental example of a stacked Moire lattice of strontium titanate recorded by Steph on our Continuum and K3 spectrometer. And what we're comparing here is the diffraction pattern at a random probe position with and without the energy slit placed in, so with and without the inelastic background. And so if we just scan around and you look at the two diffraction patterns, they look quite similar to each other. And you can see that the contrast changes as we reach different regions of the material that are tilted to different angles. But really, there's not a lot of difference between these two patterns. So in particular here, we think that because the plasmon scattering is not that strong at the location of the bright peaks, it's not really going to strongly modulate our measurements here. So if we look at the mean diffraction patterns here, they're nearly identical, but especially in the unfiltered signal, you can sort of see a bloom around the beam stop near the origin. That is the signature of this inelastic background. And if you look at some of the diffraction disks here and compare them left versus right, plotted on the same intensity scale, you can see that our intensities are shifted a little bit. But if we're making relative intensity measurements, at least in this case, it's not going to matter very much. However, if we look at this other example, this is a sample from Hem Prasad Busal and A-Ming Yan's group, also recorded by Stefan Han. Here is what it looks like, the diffraction patterns. These are fairly complicated samples. They have chromium chloride sandwiched between layers of graphene or graphite. And so there's a whole host of different diffraction spots that we measure here. So if we put down a couple of virtual detectors, the blue on the innermost ring and red on the second ring of diffraction spots, the innermost ring corresponds to chromium chloride, and then the second ring corresponds to the two different graphene lattices here. And you can see some sort of stacking defect here. So in this case, what we want to do for every probe position is do quantitative fitting of the diffraction spot intensities and positions. And in particular, we're after the chromium chloride structure on this innermost ring of spots. So if we look at that innermost ring of spots, this is how we can figure out what the stacking of the chromium chloride looks like. So here's what a virtual detector looks like, where 2 of the 6 innermost spots have been selected. So this is the first monoclinic ordering. If I rotate 60 degrees, the second monoclinic ordering, rotate 120 degrees, the virtual detector, the third monoclinic ordering. And if all 3 peaks are equal intensity, this is the hexagonal phase that essentially has no monoclinic distortions or intensity variations here. So in other words, if we can measure the intensity of the innermost ring of these spots, we can estimate at every probe position what the local stacking sequence is. So how does this look? Well, what we do is we take our diffraction patterns and we do whole pattern fitting, where we're essentially constructing a model that includes the background, all the different lattice positions, the Bragg disk shapes and intensities, and then we fit it to the mean diffraction pattern, shown on the upper right here, comparing the mean diffraction pattern to the upper middle, which is our refined model to the average diffraction pattern. And then we repeat that procedure for every probe position. And so here's what it looks like for the unfiltered data. And you can immediately see at the origin how much worse the unenergy filtered data looks. And so if you look at high scattering angles, though, they are very, very similar. But the low scattering angles, this is the region that we're interested in this sample. So here's what the model components look like for the energy filtered and non-energy filtered signals. You can pretty much ignore the backgrounds. You can more or less ignore the graphene lattices because that's not what we're after here. But what I want to focus in on is the innermost ring of spots in the chromium chloride lattices. And you can see that the unfiltered data, 3 of the 6 innermost spots have dropped into the noise floor. And that definitely has consequences for our estimates of the structure here. So in particular, if I just pull out the numbers from monodomain regions in these 2 scans here, the specific numbers don't matter too much. But if you look at the energy filtered data and we look at the 6 intensity values, 2 of the spots, the U and negative U direction, are very bright, and the other 4 spots are much dimmer. And so this corresponds to one of the known phases, the monoclinic phase. But if you look at the peak intensities of the other sample here, 3 of the 6 values are extremely low. They've essentially fallen into the noise floor of that low scattering angle inelastic plasmon. And the standard deviations also tend to be higher in a relative sense here. So in other words, we just can't even fit these peaks without energy filtering. So the only other thing I want to mention about this sample is here we're only talking about the bright spots, the parent lattices. But if you want to include all the moire lattices, all these super dim spots that you can barely make out in this image here, this is where energy filtering is like night and day. It's the difference between even being able to really see the moire peaks or not see the moire peaks here. So another example, this is from Lillian Vogel in Andy Miner's group, silicon germanium tin. This is an unfiltered scan and an energy filtered scan. And they look quite superficially similar, but if we take a look at the 2 line traces here, shown as this black line and this red line here, and then compare them, you can see additional superlattice reflections appearing at the 1/2 order spots, and they are completely more or less invisible in the noise floor in the unfiltered data here. So once again, if we want to map the presence or absence of these superlattice peaks, we absolutely need energy filtering. Okay, so let's look at even more complex examples. So Anna mentioned 5-dimensional STEM. That's what we're doing here. So this is the same setup I've been describing here. This is from Yang Yang's work, who's now at Penn State and was in Andy Miner's group when we did this work here. So again, our unfiltered nanobeam diffraction pattern and our filtered diffraction pattern, we can compare them by just moving in the energy slit in or out of the path outside of the spectrometer, but before the K3 detector here. So what we want to be able to do is do quantitative diffraction and strain mapping on this sample. And so let me show you what that looks like. So we use our bullseye pattern STEM probes. And these are apertures that we place in the condenser plane of the microscope to stamp a pattern into the diffracted disks. And this really dramatically improves the precision and accuracy of our subpixel scattering vector length measurements, which in turn is how we measure strain here. And so if you look at the unfiltered data, it looks extremely noisy with very, very strong background signals here. And then compare this to the filtered data. It's absolutely like night and day. Most of the diffuse scattering is gone. And in particular, if you look at these bullseye apertures, they are just so much sharper and so much easier to resolve precise features if you use this energy filtering. So what I mentioned before, this is a 5-dimensional STEM experiment. This is because what Yang is doing here is in situ mechanical testing. So what they use is this push to pull MEMS chip. And that means that if you push on it with the sample holder, it transfers that momentum or that motion, into the sample as a pulling force. And so in the green outline panel, it shows that the vector arrows of the force direction here. And you can see this sort of dog bone mechanical testing sample with a slit cut into it, a notch, And this is done because this concentrates the stress in the middle of the dog bone sample. And so if we know where the material is going to fail locally or where defects are going to be introduced, such as at the edge of a crack tip, which we're simulating here, this means we can zoom in and do all our measurements right at that location. So if you compare the nanobeam electron diffraction pattern here to different properties that we want to extract here, virtual detectors for them, this is the first step. We figure out where all the lattice positions are, and we want to do lattice fitting with very precise subpixel locations. But we also want to pick up either the bulk matrix or local defects such as stacking faults and twins. And so this is just showing what the virtual detectors look like that are optimized to pick up contrast from stacking faults and twins here. And this was done by comparing high resolution data to low resolution data here. And of course, in 4D-STEM, we can map a much, much larger field of view, and we don't need to be perfectly on zone to do atomic resolution measurements here. And so with energy filtering, these signatures are very, very clear. So if you look at the original single pixel detector, you can sort of make out a somewhat defected region, but not much else in the sample. Whereas if you look at the virtual images here, you could easily pick up both twin and stacking fault contrast here just by doing virtual imaging. And in particular, in this sample, we can also do our strain mapping here. So local deformation of the film measured by the spacing between the Bragg diffraction peaks enabled by these bullseyes. And I should also mention too, these bullseye apertures we give away for free. So if you're interested in installing one into your microscope, please feel free to send me an email and we will send you free apertures to install in your TEM. Okay, so this is what the study is really all about, comparing a medium entropy alloy, in this case chromium, cobalt, nickel, to a pure FCC nickel sample. So under no load, this is what the sample looks like with the notch cut in it shown above. And then this is what it looks like under load. So 1/2 of a time step here. And so you can see in the pure nickel, a little bit of stacking fault signal has been injected here, and significantly more stacking faults in the medium entropy alloy. However, when you relax the mechanical push-to-pull holder here, essentially all of these local contrast almost disappears. So there's a little bit of residual defects present, but you more or less recover the original bulk structure here. However, if you repeat this 1,000 times, so 1,000 loading and unloading cycles in the elastic regime of this material here, now you see a big difference between the medium entropy alloy and the pure nickel. So these twins and stacking faults in between them have been injected into the material and they become irreversible here, whereas the pure nickel is essentially perfectly reversible deformation here. So in the case of this study here, which was looking at the different mechanical properties of these materials here, we are able to identify that after 1,000 cycles here, there's a significant difference in the stacking fault energy, and therefore we think it's this rejuvenation mechanism. So just a little bit more on that here, looking at comparing a virtual image of the stacking faults, and then this is just a zoomed out dark field image where you can sort of see the injection of stacking faults and dislocations here. In particular for these materials, when you have high stacking fault energy versus low stacking fault energy, you get very different behavior. So pure nickel, for example, with a very high stacking fault dislocation energy, excuse me, the mechanical defects that are introduced here are full dislocations, and it's essentially an irreversible process. But if you look at medium entropy alloys, at least initially after the first few cycles of load here, the primary plastic deformation mode is injection of partial dislocations, which is essentially reversible. However, these alloys have a lot of short range order. After you've cycled this material many, many times here, you destroy the local short range ordering, which in turn lowers the stacking fault energy, and now these defects become irreversible. And so we think that this is responsible for the vastly different mechanical properties between the medium entropy alloy and the pure FCC alloy in this case. So one other example, this is one of my favorite ones of quantitative diffraction analysis. This is a twisted bilayer graphene sample from Corbina Bediaco's group that we published on. And this work was really led by Nathaniel and Maddie Van Winkle here. And so if you look at conventional dark field imaging, you can see the moire lattice between the two graphene lattices. And this is on a boronitride substrate, by the way. So this type of work has to be done in diffraction space. And this is the 0, 1, bar 1, 0 diffraction spot. And if you rotate to the 1, 1, bar 2, 0, you see a different sort of set of constructive and destructive interference, which outlines the different sort of moire unit cells here. But of course, in 4D-STEM, we have a measurement at every probe position, and we can do quantitative intensity mapping here. And so this is just showing the different stacking sequences, either AA, AB, or the 3 saddle point energy, stacking sequences of twisted bilayer graphene. So why do we care about this? Well, if you rotate the graphene layers a very small amount from about 0.2 degrees to about 1.2 degrees here, the graphene diffraction spots overlap each other. And that's shown in the lower right here, zooming in on the twisted bilayer graphene diffraction spots. So we get an interference pattern between these overlapping spots, and that intensity corresponds to the relative shift of the lattices with respect to each other if you model it over all 6 or all 12 of the diffraction spots here. And in turn, if we have many different probe positions here, we can unwrap this and measure the sort of deformation field of our twisted bilayer graphene. And so I'm not going to go into the extreme details here other than just to say, with energy filtered diffraction, this works very well. And so we can measure the sort of expansion of the white regions, the favorable AB or BA stacking of our twisted bilayer graphene. And then in between, you can see it's bordered by saddle point stacking sequences. And the sample acts with local deformation to minimize the black regions, the AA or BB stacking, because it's extremely unfavorable. We can do this as a function of Morey twist angle here, and then identify the different relaxation mechanisms in this material. Because from these dislocation or deformation or displacement fields here, with a little bit of work, we can differentiate them, and that recovers the strain field of these materials. So the relative deformation of the 2 graphene sheets with respect to each other. So that's all I'm going to show today. If you want to know more information about 4D-STEM, I wrote a review paper. And I've also written a more recent review paper covering all of STEM, including imaging, diffraction, spectroscopy, and tomography that was just published this year. Our Pi4D-STEM code can be found on GitHub along with many different tutorials. And what Steph is going to talk about is iterative phase retrieval algorithms, and especially how energy filtered diffraction can enable them. And we have a new preprint on the archive too, if you want to read about this. And so the last thing I'm just going to show is this photograph from our tutorial workshop. This was at Microscopy and Microanalysis this year. We organize these workshops all the time at different conferences. And so if you're interested in 4D-STEM analysis, please sign up for one of these. And Pi4D-STEM even won an R&D 100 award this year, which we're very excited about. So with that, I think I will hand it over to Steph.
Thank you very much, Colin.
Awesome. Okay, great. Yeah, so I guess, as I was saying, I'm going to be talking about something a little bit different. In the experiments that Colin showed, we have a large probe in real space and large separation of disks. And we're using the information of where those bragg dislocations are to learn about our sample. But instead, I'm going to be talking about experiments where generally you want to have a lot of overlap between your disks and a small probe in real space, and you're going to be using this for iterative phase retrieval experiments, so recovering the phase of your sample. And I'll talk about this in more detail, but on my title slide here in the top row, I have a set of diffraction patterns that are not energy filtered, and just below that, a very similar region where I took data that was energy filtered, and you can see kind of the difference in signal to noise between these 2 measurements. So this is, I guess, it's sort of a spoiler, but you can see why we might like to use energy filtering for some of these experiments. And I will come back to this, but I'm going to start by talking about what is phase retrieval and what we're trying to do here. So what we want to do is solve what is the phase problem. So in a 4D-STEM experiment, or any STEM experiment, we have an initial probe. This is a complex wave, and it's going to interact with our sample, which has some transmission function, and we get this exit wave. And then the problem is right here, is that our detectors are going to record the intensity of our exit wave, and this phase information is lost. So anything we can do to recover the phase information is really great. We get dose efficient imaging, so we're making more out of the same dose. And we also can see our heavy and light elements at the same time. And there's a lot of ways that you can recover the phase information of your sample, but 4D-STEM offers a few different opportunities and ways that you can do this. And I'll be talking about this in the presentation today. So depending on how you take your data and your sample and what you're sort of trying to learn about your sample, you end up with a 4D-STEM data set and a set of diffraction patterns. And as I mentioned, there are a few ways that you can recover the phase of your sample, and I'll be talking about these today. So the first is going to be differential phase contrast, or DPC, which is sort of the simplest and most straightforward. Then you can do a parallax measurement, which is slightly more complex and slightly more computationally intensive. And then the last is going to be tychography, which is certainly the most challenging. I hope one thing that you see in the presentation today is I actually like to kind of use all of these together. So I will start with DPC and parallax before moving to tychography, because you really need careful calibrations and to understand your kind of your sample and your data well. And DPC and parallax can provide a lot of that information. And these methods are not just for our research, we put them in Pi4D-STEM. And also, as Colin mentioned, we have recently put on the archive a preprint kind of covering these methods in more detail. So I'll start by talking about differential phase contrast and DPC. And to talk about DPC, you need to talk about center of mass. And I'll be showing an example here. This is the data set from the Muller group. It's a twisted bilayer of MOS2. And this is the dark field image. And we will improve upon this with our phase reconstructions. So in order to do DPC, as I mentioned, you first need to measure the center of mass. And when you have your STEM probe, it's going to interact with your sample potential, and it's going to shift, or you're going to redistribute the intensity, or you might see a rigid disk shift. And this, you can measure this with some sort of first moment detector and measure your center of mass. And you do this for each probe position, you get your center of mass X and your center of mass Y. Now, one thing is you're not going to have necessarily good rotational alignment between your real space and reciprocal space. And there's going to be some rotational offset that you need to calibrate. And you need to calibrate this for a high quality DPC reconstruction. And this is even more important when you go to tychography. You're going to want to know this angle very well. And one thing you see here in these images is your X and your Y are not perfectly aligned. They're on the diagonal. And we're going to fix this. So what you can do is rotate your reciprocal space relative to your real space. And you want to have a conserved vector field, or kind of minimize the curl or maximize the divergence, maximize the signal in your DPC, however you want to think about it. In this case, we are minimizing the curl, and we've solved for our rotation here. It's about 27 degrees, and we can fix our center of mass. So now it's looks much more like it's in X and in Y. One other thing I'll note is, as you're kind of doing this with your own data, this is really helpful for solving the rotation, but there's still a 180 degree ambiguity. And you can solve this by making sure that you have the right phase shift for your sample. It should be a positive phase shift. So now we can take these center of mass measurements, which we've calibrated, and we can get the phase of our sample. So center of mass is related to the gradient of our DPC reconstruction, which is proportional to the phase of our sample. So we can do just a little bit of algebra here, and we can get really nicely a phase reconstruction of our sample. And DPC is really good. This was fast. This was pretty easy. We just had to do very little calibration, and we get this nice reconstruction. You'll see in a minute the challenge with DPC, though, is whatever aberrations we have in our probe, in which you may have defocus. We'll see why you might want to add defocus in your probe, and that's going to be transferred to your sample. So DPC isn't as good in this case. So this is a different data set that I acquired. And in this case, I have a high sampling in real space. So my step size is about 2 angstrom. And I also have quite a bit of defocus. So you can see now why dark field and DPC are not so good. I'm not able to see really any of the fine features that you'd like to be able to see in a gold nanoparticle sample. And DPC does, though, provide some information and helps us solve this rotation, which we measure as 13 degrees. But DPC really works best and excels for in-focus data sets. So I'm going to introduce a different method that can be really helpful for solving your defocus and also your rotation and providing these sorts of phase reconstructions. And this is going to be the parallax approach. So if you imagine here, this green dot is going to be a virtual detector. We're just taking one spot in the center of our diffraction pattern here, and we're going to see the virtual image. And now I'll play this movie, and I want you to see how these virtual images change as we move around the central disk here. And you can see that we see a lot of shifting of our diffraction of our gold nanoparticles, and this is getting more and more pronounced as we move further from the center. And this is basically the parallax effect. Each of these virtual diffraction patterns can be thought of as an incoming plane wave at a different angle. And when we align all of these images, and what we're going to do is align all of these images. So you can see here, I guess it'll play the movie again. And we have now created a vector field where we have taken all of these virtual images and done a cross correlation to align them, and we get this vector field that looks like this. So we can compare, this is really helpful. We can get a rotation, and we can compare this to our DPC. This is in good agreement here, the rotation between our DPC and our parallax, which is a good sign. And we also now can back out the defocus and other aberrations by fitting an aberration surface to this vector field. And you can see how this looks now. We have a parallax corrected image where we've basically taken all of these low signal to noise virtual images and align them. And you get, you can start to see now some features in the nanoparticles that you couldn't see before. And we also know what the CTF is because we fit this aberration surface, so we can do a parallax correction. We can go even a step further. We can do some subpixel alignment and upsample the bright field disks. And now you can see really nicely, you can even see some gold, some lattice in these gold nanoparticles. So we're getting resolution here that is higher than our sampling of the data that we took in real space. So parallax is a nice way to do a phase reconstruction of our sample and sort of a medium amount of computation, but we can go even a step further, and for that, we're going to be doing tychography. So I'll explain a little bit about how tychography works. We're going to be solving for our probe and our sample potential. So what you do is you start for a guess for our initial probe and our initial sample potential, and this is where the calibrations that I talked about are really important. And you compare this to your experimental diffraction patterns. And then you're going to update your guess. And you're going to do this for each probe position for multiple cycles. And there are a lot of advantages to tychography. So one is if you have sufficient dose and you can get super resolution imaging. And this is because your resolution is set by the maximum scattering angle that you acquire. So you can get information beyond your nominal central disk. The second thing is you can do aberration correction, which is really helpful for helping remove aberrations in your probe, and also provides a nice benefit for us, which is that we often like to add defocus. So if you see over here, if you have an in-focus beam and you have a lot of overlap, it's going to be focused to a very fine point. So if you want to cover your whole sample, you need to take a lot of steps. However, if you're defocused, now you have a much bigger probe. So what you can do is you can take larger steps in real space to cover the same area like this, and you have some overlap between your adjacent positions, and you can cover your field of view like this. So this is really helpful for us for both dose fractionation and also for sort of faster data acquisition. There are some challenges with tychography, as I mentioned. It's more challenging, more computationally intensive, and you're also not guaranteed to converge. And this is where regularization can be really helpful. So just to further help clarify, let me show you again what this experimental geometry is going to look like. We're going to be scanning our probe back and forth, and you're going to be defocused. So you see sort of a shadow image. You can see your shadow image of the sample here in the Rankine-gram. So how this is going to work is we're going to start with initial probe that looks like this. Again, this comes from our defocus that we've measured from parallax. This is why it's really nice to do the parallax before the tychography. And we also know our convergence angle and we know our energy. We start for our guess for their object phase. And then we can also put where we take all these 4D-STEM measurements, fill in these red spots in this position like this. And we can simulate what happens here in this overlap. And we get an image that looks like this. We can compare this simulated diffraction pattern to our experimental diffraction pattern. And you will see that these images are not the same. And that's okay, because we can update now. And we're going to do this for a number of cycles. So what we can do is we can slowly start to update, and you will see that we will move towards having our simulation match our experimental data. And we have this nice reconstruction here now of our MOS2. And if you compare all of these, you can see that tychography is the best. We have the highest resolution in this case. But this is for an in-focus data set. So DPC is also doing pretty well. And parallax also provides a nice space image. So kind of the approach that you want to use is going to depend on your sample, your measurement, and what you're trying to learn. As Colin mentioned, this is part of Pi4D-STEM. We have tutorials, we have our code. You can run it in Colab. And we have a lot of other features for tychography that is in this code. So one thing you can do is mix state. So if you want to help solve the problem of coherence, you can work with multiple probes. We have multi-slice reconstruction. So this is to help solve phase wrapping problems. You can break your sample into smaller slices. And we also have a lot of regularization, which is really important for helping get your reconstructions to converge. And this is here showing an example of how we regularize the probe. which will lead me to say that one thing that's really helpful for probe regularization is to acquire a vacuum probe, because you can put some more constraints on the problem that way. And also for the type of experiments that Colin showed as well, a vacuum probe can be really helpful for helping make better measurements and reconstructions of your sample. So then, excuse me, that I've introduced tychography, I'd like to kind of move back to the main topic, which is energy filtering. So these are the same diffraction patterns that I had on my title slide. And you can see as you go across the gold nanoparticle, you can see the lattice, and you see this is the unfiltered data. And then now I'll show the filtered data by comparison, and these diffraction patterns are plotted on the same scale. So now that you understand better how these sorts of phase retrieval algorithms work, you can see why it might be really helpful to remove some of the unwanted scattering and background signal to get these nicer diffraction patterns here that are filtered. You see more information beyond this nominal central disk, and you also see more signal to noise in these patterns as well. So this is a lot clearer. And now we can see what happens when we actually take this data and go towards our reconstructions, even though we have a suspicion looking here that these lower diffraction patterns are better. And we're starting with the unfiltered case, we can do our dark field reconstruction. We don't expect this to be very good because of our defocus and our real space sampling. We can do our parallax, and this is a nice reconstruction where we can start to see some of the features in the lattice spacing in our parallax. And then tychography, which is better. We see higher resolution reconstruction. But one of the things that I don't like so much about this particular example is that you're not converging while you're low spatial frequencies. And this makes sense given kind of the unwanted background signal and the types of spatial frequencies we'd expect that to, where we'd expect that to be. We can compare this to our filtered diffraction patterns now, and we can see our dark field here. Our parallax is perhaps slightly better, but it's hard to make a very totally fair comparison because there are different areas and different particles. But with tychography now, we can see really nicely with this last diffraction or last reconstruction over here, we are converging the high spatial frequencies, very similar to the unfiltered in terms of our resolution that we can achieve. But now we've been able to converge our low spatial frequencies as well and get this really nice reconstruction of our gold nanoparticle. So I'll blow this up so you can see this even more clearly. Both of these have sort of similar resolutions in our reconstruction, similar features that you can see, but because we're doing this energy filtering, it's going to help us converge our lower spatial frequencies and get this even nicer reconstruction. So I've talked a lot about some different phase retrieval methods in using 4D-STEM and how energy filtering can be helpful for some of these. And that's what I have. We'd be happy to take questions.
Thank you very much, Stephanie and Colin, for amazing presentations and great results. We have a little bit of time for some questions, so maybe I'll just start from the top. So Colin, someone is asking if there is any point to measuring diffraction patterns with the slit centered on the plasmon peak.
It depends on the experiment. So it depends on the property that you're after. If you are trying to do like the type of measurements that I showed in that 5D in situ strain mapping, where you want the elastic signal, then there's not really a big point. You kind of just want to use your slit to focus or to remove all the plasmon scattering. However, especially at the beginning of my talk, I showed some examples of momentum energy combined mapping, where you actually want to be able to trace out all of the different diffraction vectors, both in momentum space and energy space, to see what the allowed states or bands of the material are here for scattering. So it really depends on the property being measured that you're interested in.
Thank you. Just because you mentioned the 5D experiment, someone else is asking, did you have a script to acquire the data, or are you collecting those 5D STEM data sets manually, moving the slit and changing filter mode, etc.?
For the 5D experiments, those also involve in situ mechanical holders, and those in situ experiments are really hero experiments. They tend to be extremely complex. And so the typical way of doing it will be, we'll set up all of the microscope parameters on the spectrometer and the diffraction, camera length, semi-angle, using a bullseye probe, etc., and try not to change those settings. So they sort of stay constant. And then, it's a semi-manual procedure because you really want to synchronize your data collection to your holder, manipulation of the sample, in this case with the push to pull in situ mechanical cycling. And so it'd be nice to sort of script control absolutely everything, but often something unexpected will happen, or you'll have to realign your microscope, realign your pattern, and that means that you need human intervention anyways. And so, until we have full Python scripting control of the holders, the microscope, the detectors, everything together, it's going to be very challenging to fully automate those experiments. And I still don't think it'll happen because you need a human to fix any errors that happen.
So if I may add a little bit, the experiment that I showed at the beginning of the presentation, where we were doing 4D-STEM EELS, EDS, and heating experiment, that was a Python scripted, so that was collected overnight, automatically controlling the temperature on the holder because the holder is integrated in digital micrograph, and then switching modes on the filter between FDEM and EELS mode and collecting spectrum image data sets at each point. So as you said, Colin, I think it depends on how much communication there is between different hardware, microscope, the detectors, and the holder, and integration would help a lot if you were to do a 5D-STEM experiment, which is a hero experiment, as you said.
I'm actually impressed. I'm very impressed that was automated. That's so cool. I did not know that. You should highlight that.
Yes. So that was a script that Liam wrote, and we do have the script, and we're happy to share it with people if they're interested. Stephanie, maybe a question for you. What kind of samples benefit from energy filtering for phase retrieval?
Yeah, I think it's going to depend on how much unwanted background scattering that you have. So again, I think thickness is going to be one of the main determinants, but also just in general, how much plasmons and what you expect from your sample. So some samples will have a lot more background scattering than others. But in general, I think thickness would be the main determinant.
Okay. And another question, Colin, great talk, thank you. The question is, how did you simulate the plasma contribution to diffraction?
In the laziest way possible. So plasmons essentially have a Lorentzian distribution. And so if you just pick a characteristic energy and figure out what cross-section you want to use, you can compute the Lorentz distribution and then either convolve it with your pattern, which is what I should have done, or just add it to the pattern, which is what I did do. And I added it because all I wanted to do was see what happens at low scattering angles to test just the simplest possible thing. If I'd convolved it with my pattern, the results would have been even worse. So I might actually do that in the future. I should mention there are some new ways of simulating plasmons more accurately. In particular, there's a paper from Boudica Mendes from this year that shows a Monte Carlo method, an incoherent phase method that we figured out to do better plasmon simulations. But in this case, all I wanted to do was show what background does to the simulation.
A very interesting question just came in. Have you guys explored energy filtered 4D STEM with biological samples?
F?
I personally have not, but I think it would be really, depending on the sample, could be really helpful because these tend to be thicker as long as it wasn't too thick. I don't know, Colin, if you have anything else that you'd like to add.
I just don't think there's many. I mean, so energy filtering is typically used in biological experiments for HRTEM, for example, on the standard Krios setup with the Selectress in our energy filter. I think energy filtering helps, but one of the advantages of the methods that Steph showed, the momentum resolved phase retrieval, is it's a lot more resilient to inelastic scattering. So I think Steph is correct. It's really thick. There could be a lot of plasmons or charging, background artifacts, whatever, from the ice. But at the same time, these materials, biological samples, scatter extremely weakly. And so at small scattering angles, where the plasmons are concentrated, you're going to get some artifacts. But at the high scattering angles, where the sort of atomic scale structures or like adjacent alpha helici or protein sort of length scales, I don't think plasmons in STEM mode will hurt you very much, I guess. So it'd be very interesting to test, though, that there's not many microscopes that are set up to do these experiments yet.
Okay, in the interest of time, I would like to thank everyone who attended our webinar. Thank you again, Colin and Stephanie, for accepting our invitation and presenting these really amazing results. This webinar is recorded. We will share the presentation to everyone who had registered, and any question that we didn't have time to answer online, we will make sure to reach out to you guys and send you the answer to the questions. Thanks again, everyone. Have a wonderful day.